Hello, hello students. Good morning. I am Dr. Tejendra again before you with a new video lecture. Today I am going to discuss you some important short questions from Unit 3, 4, 7, 8 and 9 of the Evolutionary Biology, which were asked in the previous year, and hope you will enjoy as well. So this is the first question, first term. This is Coprolite, and it was asked in the year 2019. So you had to answer in a simple one line or in for the 1 mark. Okay. So what is the coprolite? Coprolite are actually fossils. These are trace fossils. Okay. So trace fossils means which were not containing the body exactly. Okay. So there are actually two type of fossils. One is body fossils and another one is trace fossils. Body fossils contain any body part like bone or any body part or complete body part. Okay. But trace fossils does not contain any body part. They contain some impressions or maybe some another products which are released from the body like fecal matter. Okay. So coprolites, as word copro means fecal matter. Okay. And light word is generally used for the fossils and especially for the rocks. Okay, so that's why it is called as coprolite, and it is also called as coproliths. So coprolith or coprolites are trace fossils, and these are fossilized ficus of animals, and these are actually not a body, as I told you. And this give a clue. These give us some clue about the behavior of animal. How they give us clue? Can you imagine? Because actually they these are some fecal matter. It means these contain the undigested food. So that's why they also contain some material which gives some information to us that what they had ate. Okay, like suppose any animal was carnivore at that time and you got a fecal matter, so how could you identify? Because carnivore generally ate animal directly, and that's why if the animal was a mammal, it means in the fecal matter their hair will be present, right? So this is some type of identification generally used by the some environmentalists and some scientists. So any another type of like how their rotation is like. Okay, and how their formation structure, and formation is also based on the structure of the alimentary canal. So that's why coprolites are used to to find some clues, to some hint about the diet behavior of the animals. Next important term is bootstrap value. What is the bootstrapping value? Bootstrapping value is a value which we used for the determination or to identification of the phylogenetic tree. Okay, so phylogenetic, whenever you make some phylogenetic tree, then you put some values on their branches. Okay, so suppose that branch is uh, a partitioning branch, then you give the 0 number, or suppose it is not a partitioning branch, then you give the 1 number, and so on. Okay, so this is the type how phylogenetic tree is formed, and that's why some bootstrap value give us the some idea. Actually 0 and 1 are not bootstrap value. Bootstrap value is like percentage, it is based on the 100. It means that how much your value is on the based of the 100, means out of the 100 how much your value is. And it tests reliability of the inferred tree, as whenever you are making a phylogenetic tree, and if suppose you have m number of sequences and n number of nucleotide or codon or maybe amino acid also, and you form some branches and you count the value for the branches. Okay, so if you get some value like 90%, 95% or more than 95%, then it means it is correct. Otherwise it is not exactly correct, or not it is resembling. Okay, so this is the bootstrap value, which gives some idea about the some branches which were used in the phylogenetic tree. Next question is for differentiating between the body fossils and trace fossils. As I told you in the first slide, so body fossils contain body parts, but trace fossils does not contain body part. Okay, so and again, body fossils have means it may be organ, it may be tissue that are fossilized, that are in fossilized form. Okay, but trace fossils have contains fecal matter, sometimes footprint also. Okay, one example of the trace fossils like dinosaurs foot mark. You might heard sometimes in the newspaper or sometimes in the bulletins that there are some footprint mark were discovered of the dinosaur. So these are actually trace fossils. Okay, body fossils also give us information about the anatomy and morphology, as I told you in the first slide, that how the fecal matter is constructed or how its structure is there. It gives some information about their diet behavior. Okay, so trace fossil give us diet behavior, but body fossils give us the anatomical and morphological structure and information about that. Okay, so that exact information is received by the, or it gained by the body fossils. Okay, right, like you have find some bony fossils or bony structure in the fossil, then you can easily say that okay, this animal was like that at that time. But if you find a trace fossils, then you only just imagine. Okay, but there is no clear cut evidence that how that animal was. Okay, again, differentiation between the autopolyploidy and allopolyploidy. Autopolyploidy, auto means itself and poly means many number. Ploidy is denotation mark or word used for the our chromosome number. Okay, so whenever its number is higher or lower, then it is also used as ploidy. And there are many types of ploidies there: polyploidy, aneuploidy, euploidy. Okay, you might learn it in the previous classes, so it is very common term for you. Allopolyploidy, allo means different. Okay, so autopolyploidy and allopolyploidy. Autopolyploidy and allopolyploidy both are depends on the chromosome of the same species or different species. If they are from the same species, then it means it shows autopolyploidy, whereas from the different species it shows the allopolyploidy. Okay, so if there are more number of chromosomes present in a species and they are received from the same species, then it is autopolyploidy. And whether it is received from the different species, then it means these are allopolyploidy. Next difference is homologous chromosome Homologous chromosome is what? Any person or any individual have uh, two copies of chromosome in their body, in their cells. Okay, one set of chromosome is received from the paternal and received from the father, whereas second set of chromosome is received from mother, and that is called as maternal. Okay, so these two same chromosome, like I am talking about the 25th number chromosome, so it means there is also available a different 25th number chromosome is there, and that 25th is may be received by the mother or father. It means it depends on that which parent you are comparing. Okay, so one is received from the father and another one is received from the mother. So these are the homologous chromosome, but remind it that these both were same in their genetic structure or gene alignment. Okay, so these are homologous chromosome. So autopolyploidy is generated by the homologous chromosome. You might now understand that why homologous chromosome, because it is present in the same organism, same cell. But allopolyploidy is not generated by the homologous chromosome, because these are not present in the same organism. These are present in the different organism. That's why it is not created by or not generated by the allopolyploidy. Next difference is non-disjunction of chromosome, and this is a very peculiar feature for the crossing over, and you might learn it in the genetics unit. So non-disjunction of chromosome creates or generates autopolyploidy, whereas mating with the different species or different organism of the different species, that generate allopolyploidy. Now examples. So autopolyploidy is generally found in the plants, animals. I don't know actually, and it is generally saying that rarely or sometimes not present. But allopolyploidy is their example, like mule. Okay, mule have actually 63 chromosome out 63 chromosome, and these 63 which they were received from both ass and horse, and one set contain 31 and another one contain 32 chromosome, and that's why their overall number is different, means 63. So this is actually different, and that's why they are infertile. Okay, so in case of animals, ploidy condition means polyploidy, like autopolyploidy or allopolyploidy, is producing infertile progeny, and these are not viable. But in case of plants, there are some beneficial, sometimes beneficial, but in case of animals, it is not beneficial. Now justify. This was question asked for the justifying in the question paper, and this question was: molecular data can be used to predict evolutionary relationship. So if you are attending the questions of justifying, then you have to mention answer in yes or no first and then explain. Okay, so yes, this molecular data can be used to predict evolutionary relationship, right? Then give the explanation why. Because molecular data is also using for the forming or generating phylogenetic tree, we know very well. And their example is like rRNA. rRNA is also used mostly for the relations to identifying or to gathering some information and to identifying the similarity between the species, because they have most conserved sequences. And one another thing is repeated sequence. As you know very well that whenever a species get evolved and it generates some new species, okay, then that time some set of genes is repeating over the evolution and over the new speciation, and these repeating sequences are present in the cell, and these are called as repeated sequences. On the basis of these repeated sequences, we generally identify that how we are similar from our ancestor, and how any species are different from their species, their another ancestor or neighboring species. So they show us the relatedness or relationship between those, and on the basis of these, ortholog, paralog are also identifying. So these molecular data, of course, can be used for the predicting evolutionary relationship. Next, this question was asked for the fill in the blanks, and that question was the software is used to construct phylogenetic tree using molecular sequences. So although there are more number of software are present at the present time, as my estimate, there are, I think, more than 60 or 70 software are present till the time for the making phylogenetic tree. Most of them are paid version, and sometimes these are some are free also. As I know, Mega is the best because it is, first of all, it is free, and we also prefer that free software. So it is, Mega is free. And what is the Mega? Mega is the molecular evolutionary genetics analysis. It is the complete full form of this. Okay, and it is also evaluate and use for the maximum composite likelihood method. Okay, so this uses, and there are also uses some cluster W, and some detailed information are also available on the some internet resources you can find there. And how could you use this is there are some also tutorial also, but this question was only asked you for the some only for the exam and for the fill in the blanks, so I'm not giving some detailed information about it. Okay, so our lecture is completed. Actually, this was the part first, and I also make some another some second part for the some another questions. Which question I would take for the next part? Please give me some feedback and some suggestions so that I understand that which question I will take. Actually, I took these short questions because at the present time, you know that UGC UGC released a notice, and they said that short examination should be there, like 2 hour or instead of the 3 hour. So I think it might be an online exam will be there, so I think short questions will be there, and it might be MCQ or it might be some short questions. So it let's see what happening. As per university guideline, we will think about it, but as my assumption, so that's why I took yet only short questions. But if you want a detailed explanation on the larger and descriptive type questions, then you can also comment below on this video. Hope so you will enjoy, and best of luck for your preparation. Thank you.